In this video, I'm going to discuss section 3 of chapter 24. In this section, we're going to go into what happens when there are different numbers of chromosomes other than the usual number. So normally, as you can see in this picture, we have a karyotype. It's a process in which chromosomes in a cell can be arranged in pairs according to their size and general appearance. So as you can see here, we have a typical normal human with 23 pairs of chromosomes. 22 of those pairs are called autosomes. Those are the regular non-sex chromosomes. And in each pair, the chromosomes are said to be homologous. So we have a homologous pair of chromosomes, for example, for chromosome number 1, same thing for 2. Note that when we arrange chromosomes in this way, we start with the biggest chromosome, number 1, so the longest chromosome, which has the most genes. Then we go to 2, 3, 4, 5, 6, 7, and so forth. You can see the chromosomes are progressively getting smaller and smaller until we get to chromosome number 22, which is very small. That's the smallest autosome. And then finally, we have one last pair of chromosomes, which are the sex chromosomes. In this particular case, this is a karyotype of an individual who's a male because there's an X chromosome and a Y chromosome. If there had been an X and an X chromosome, then this would be considered to be a female. So karyotype information is very useful. It can help us to determine if there are any chromosomal number abnormalities. In this slide, I'd like to talk a little bit about how a karyotype is made. You have to start first with obtaining a cell sample. In an adult, this is relatively easy. You would just get a blood sample, and within that blood sample, there would be some white blood cells, and then you'd sort of separate those out, and then you would use those as your sample cells. In a fetus, it's a little more complicated. There's one way to get fetal cells, which is through getting their fetal cells with the amniotic fluid, which is within the amniotic cavity. So if you would just withdraw some fluid, you will get some fetal cells, and you can look at those. Another way you can do this is to obtain cells from the placenta. Okay, and that's shown here also. Let me see if I can point here. Let me see if I'll get my pointer here. So over here, you can see the amniocentesis, which is the first method I talked about, where you get some amniotic fluid, which would contain some fetal cells. Here's the second method in which you get cells from the placenta. Now, it turns out cells from the placenta contain the same genetic material as the fetus, and so that's another way of doing it. So they're zooming in here to show you what's called the chorionic villus. What they're going to do is get some cells from this villus, and then do a karyotype analysis. So once you have the cells, the next thing is to look at your sample of cells. When you see cells that are dividing, you want to stop them from dividing by adding a toxin. So you stop it just when the chromosomes are at their most compact, which would be, say, late prophase, around metaphase, something like that. The chromosomes are then stained and then photographed and arranged using a computer. Now that we've talked about how a karyotype is made, let's talk about some of the things that can go wrong that can lead to an abnormal number of chromosomes. So the general name for this sort of phenomenon is called non-disjunction. So this is a failure of chromosomes or sister chromatids to separate either during meiosis 1 or meiosis 2. So recall there are two phases of meiosis, 1 and 2. In meiosis 1, homologous chromosomes separate, but each homologous chromosome consists of sister chromatids. And then in meiosis 2, the sister chromatids separate. Okay, so non-disjunction can occur during meiosis 1 when both members of a homologous pair go to the same daughter cell. So if both members go to one cell, that means one daughter cell will have too many chromosomes, the other won't have enough. Or it can take place during meiosis 2 when the sister chromatids fail to separate and both daughter chromosomes go to the same gamete, leaving, again, one gamete with too many chromosomes and another with not enough. Some resulting gametes then have the incorrect number of chromosomes. So this can result in trisomy or monosomy. So trisomy is when you have a chromosome that's present in 3 copies, and monosomy is a chromosome that is present in 1 copy. In the next slide, we're going to look at a picture, a diagram explaining all this. Okay, we're going to go through and look at the 2 types of disjunction that can possibly occur. Recall that one type of disjunction occurs during meiosis 1, and the other type of disjunction, non-disjunction, occurs in meiosis 2. So what we're going to assume here is that the sperm are generated normally. Of course, potentially the sperm could also have a problem, or and so forth. So we're just using this as an example. Let's look at non-disjunction here. We'll look first in the case of non-disjunction in meiosis 1. So we're just showing a pair of homologous chromosomes. Obviously, there are 23 pairs, so we're just showing 1 pair here. So let's assume these are autosomes just for sake of argument, although they could be sex chromosomes. Okay, so here we have non-disjunction event in which we have two homologous chromosomes. They should separate in meiosis 1, but they both go to 1 daughter cell. And so that leaves 1 daughter cell with essentially too many chromosomes, and then another daughter cell with not enough chromosomes. Let's look over here at the other side. We can see here what happens normally. So if meiosis 1 goes normally, then these homologous chromosomes should be separate. Now, you might ask, why are these doubled up? Remember, these are pairs of sister chromosomes. Each chromosome actually consists of a pair of sister chromatids. Okay. All right, so let's continue over here. Now we go to meiosis 2. So this cell here divides, and this one here divides. And we can see now we have a continuation where we essentially have too many chromosomes over here. So the cells have at this point separated, but you have, I mean, the DNA has separated, but you still have too much DNA here. This normally in meiosis 2, the sister chromatids would separate. So presumably that's what's happened here. But because we started with 2 chromosomes, you have 2 chromosomes here and 2 chromosomes here. Now, if we look over here, we are completely missing this particular chromosome, so it would be completely missing. Okay, so then we have a fertilization event. So here's the sperm. The sperm has gone through meiosis 1 and 2 normally, so it has a single copy of this particular chromosome. And now there's fertilization. Now we have the zygote. So you can see here we have 3 copies of this 1 chromosome. 1 from the male gamete, which is from the sperm, and 2 from the oocyte, which is the female gamete. Here we have again 3 chromosomes. Here we just have the chromosome from the male. Okay, so this individual here would have not 46 chromosomes, which would be the normal 2n number, but 46 plus 1, so 47 chromosomes altogether. Same thing here. 2n refers to the number 46 for humans, plus 1 would be 47. Here we have, we're missing 1, so this would be 2n minus 1. So this individual would have 45 chromosomes, so would this person. Okay, now let's take a look at what happens if non-disjunction occurs a little bit later in meiosis 2. So we have this cell, mother cell, it divides into 2 daughter cells, and everything goes normally. The homologous chromosomes separate. Now in meiosis 2, the sister chromatids should separate, and that's in fact happened normally here, so that's good. Here we have a non-disjunction event where both sister chromatids remain and go to 1 daughter cell, leaving this daughter cell over here with no chromosomes, and then this one with too many. Okay, we've assumed that the sperm divided normally. Went through meiosis 1 and 2 normally. So then we have fertilization, and we can see what we get. Here we have a normal cell. It's got the 2n number, so 46 chromosomes. Here again, we have another one that's normal, 46 chromosome. Here we have a trisomy event. Meaning we have 3 identical 3 of 1 particular type of chromosome. So it's 2n plus 1. So So this means it's 46 plus 1, so 47 chromosomes in total. Here we have the female didn't contribute any of this particular chromosome, so there's just one copy from the male. So this is a monosomy. And so the number would be 2n minus 1. So it's 46 minus 1, so 45 chromosomes altogether. So normal development depends on exactly 2 of each kind of chromosome. But of course, as we just saw in the previous slide, trisomies and monosomies can occur. In general, trisomies are a little bit better tolerated than monosomies. However, in general, even monosomies lead to miscarriage. So most chromosomal trisomies lead to miscarriages. So for example, we looked at the karyotype, chromosome number 1 is the longest chromosome, and so it has the most genes. Trisomy 1 is a very rare type of trisomy that always ends in a miscarriage, and it has been documented, but they're rare because if you think about it, that chromosome 1 has so many genes on it. So if you have an extra copy of the chromosome, that's going to essentially mess things up, and therefore things will go wrong very early in development of the fetus. And there are the only trisomy that actually has a reasonably good chance of survival going through gestation and then birth and then into childhood and even adulthood is Down syndrome, and this is trisomy 21. So if you look back at the slide I showed you earlier, I mentioned how the chromosomes were getting smaller and smaller and smaller as they increased in number. That's just the way we arrange them. And so chromosome 21 is one of the smallest chromosomes. So it's perhaps not surprising that trisomy 21 is actually one that can actually survive and do okay. It has a relatively small number of genes. There are also trisomies 13 and 18. These do occur, but these individuals rarely survive past the first few months of life. So I mentioned that trisomy 21, also known as Down syndrome, is the most common type of autosomal trisomy in which the individual actually is able to survive through fetal development. In other words, through gestation, through birth, and then into childhood and adulthood. So let's talk a little bit about some of the features of Down syndrome. So Down syndrome individuals have short stature. They have an eyelid fold, flat face, stubby fingers, a round head, a single palmar crease. There's actually a picture of this coming up, but this means if you look at your palm, there's just a single crease. Most people have 2 creases, although about 1 in 30 people have a single crease. So having a single crease doesn't mean you have Down syndrome. And then Down syndrome people have mild to severe mental impairment. So the chances of having Down syndrome increase pretty dramatically, starting at about age 35, if you look at maternal age. Having said that, the extra chromosome can occur in either the egg or the sperm. It's just that it's more likely to occur in the egg than in sperm. So about 3/4 to even a little bit more than 3/4 of the time it's because of the problem with the egg. Some symptoms may be due to an extra copy of a GART gene in the long arm of chromosome 21. Okay, so what do we mean by long arm? Let me see if I can draw a picture. So if we look at a chromosome, there'll be a centromere. So that's the centromere. So this is the long arm. So there's sort of a constriction in the chromosome right at the area of the centromere. So this would the longer bit would be the long arm, and the shorter bit is the short arm. So this GART gene is located on the long arm of chromosome 21. It causes a high level of purines. Purines are nitrogenous bases such as adenine and guanine. So it causes high levels of these purines in the blood, and this seems to lead to mild to severe mental impairment. Diagnosis of Down syndrome is possible via karyotyping, which I discussed. So you could do it when the fetus is developing. So I just wanted to continue a little bit more with Down syndrome. So Down syndrome, or DS, is the most common genetic cause of developmental delay, with an incidence about 1 in 800 live births, and is the predominant reason why women choose to undergo invasive prenatal diagnosis. So I'm just reading from this paper here. I have the source down at the bottom there. However, as invasive tests are associated with around a 1% risk of miscarriage, new non-invasive tests have long been sought after. So the point of this paper that I'm referencing here is that they're looking to try and find less invasive methods of testing for Down syndrome. For example, there are some where you would take maternal blood and you might be able to figure it out from that. Okay, so let's take a look at this figure here. So here we have Down syndrome live births as a percentage. And so the percentage is extremely low, up to about 35 years of age. And then there's sort of an inflection point here and it starts to take off. And it can get very high. So, for example, if a woman is 45 years of age, there's quite a high probability of having a child with Down syndrome. It's about 3%. Okay, these are just some figures about Down syndrome. You can see some of the things that we've talked about. You can see some of the facial features I talked about, short stature and so forth. I talked about the palmar crease. So in an individual who has Down syndrome, they'll typically have a single palmar crease. Most people have at least 2 or 3 creases in their palm. But of course, having a single palmar crease doesn't mean you have Down syndrome. It's also found in about 1 in 30 people have just a single palmar crease. You can see the karyotype here. So if we look here, we see that trisomy 21 means there's 3 copies of chromosome 21. You can see that it's a very small chromosome, so that's probably why individuals with 3 copies are able to live and do pretty well. And then this is the GART gene. So you can see here, this is the long arm of the chromosome. And so they're just showing the gene there. Okay, so I'm going to switch topics a little bit. We talked about a disease such as Down syndrome, which affects autosomal chromosomes. In fact, Down syndrome was the only one where the individual would sort of do well enough to go through childhood and even into adulthood. More common in terms of non-disjunction events that actually lead to individuals that are able to do okay are changes in sex chromosome number. Okay, so I need to explain a little bit why that would be. One reason why people would say an extra X chromosomes do okay is that if you look in the somatic cells of a normal female, one of the X chromosomes is randomly deactivated. This randomly deactivated X chromosome can be seen as a small dark staining structure called the Barr body, named after the person who first discovered it, in the cell nucleus. So there's a picture here. Let's take a look here. So this is a neutrophil, which is a type of white blood cell found in blood from a female patient. And you see that little bleb there, that little blue thing, that is the Barr body. So that actually contains this deactivated X chromosome within this nucleus. Note that the nucleus in a neutrophil is very characteristically has about 4 of these lobes. So it's a very unusual looking nucleus. So because these X chromosomes are natural, one of a pair of X chromosomes is naturally deactivated anyway. That makes it a little less of a problem when you say you have a female with 3 X chromosomes. Well, the extra X chromosomes will actually become deactivated and become Barr bodies anyway. So you're left with just a single X chromosome. So that's why these individuals are able to live. Now, what about if we have if you're have an individual that has an extra Y chromosome or something like that? Well, it turns out that also leads to individuals that are able to develop and live fairly normally. There are some issues that we'll talk about in a second that have to do with fertility and things like that. But in general, you would see this person as fairly normal. So, well, the Y chromosome is different. We don't have bar bodies for Y chromosomes, but the Y chromosome is relatively small. It's one of the shortest chromosomes there is. And it really just has genes that help to determine maleness. So these are called maleness determiners. These are necessary for things, for example, like the development of testes in the fetus. And so it's not as big deal if you have, say, 2 Y chromosomes. Okay, so let's take a look at some non-disjunction events that can affect sex chromosomes and look at what happens. So the first one up here is Turner syndrome. This is XO. The O here, it is written as a capital O, but you can think of it as a 0. It's a placeholder, so it means that this female just has a single X chromosome. Now, if you look at them phenotypically, these individuals are short. They do have some malformed features. For example, they have webbing on the neck. Typically, because it doesn't look very attractive, it can be dealt with fairly easily with surgery, some sort of cosmetic surgery. They have a high palate and a small jaw. Turner syndrome individuals develop normal intelligence. However, they do require hormone therapy to undergo puberty or to menstruate. So typically what happens is they will go on to artificial hormones, essentially for life, to be able to have puberty, menstruation, and so forth. Now, pregnancy is possible in these individuals, but only by in vitro fertilization and only by using a donor egg. So they can't pass their own genetics along to the next generation. They have to get an egg from some sort of donor. Now, another one of these non-disjunction events is Klinefelter syndrome. This is an individual that's XXY. And you might think, well, is this a female or a male? Because XX would typically be a female, XY is a male. Turns out just having that one Y is enough to make a male, essentially. It has those sex determiners, right, of maleness. So this is a Klinefelter syndrome individual is clearly a male. And they can have different levels of symptoms, okay? So it varies a bit. But in general, they're fairly subtle. These individuals are usually not diagnosed until they hit roughly around age 15. So they're going through puberty and so forth. So they're typically going to have, for example, low testosterone levels and things like that. It affects quite a large number of males. About 1 in 650 males are affected. So it's not terribly common. It's not terribly rare either. So at that level. Now, these individuals are typically tall. Why are they tall? Well, one reason is testosterone, which is an androgen, which essentially is a hormone that makes men, is actually one of the things that limits growth of long bones. Okay, so these individuals are a little bit tall. They may have mild mental impairment, but of course that's not always the case. They're normally sterile, but they can father children by assisted reproduction. So, for example, it is possible. Typically, their testes do not produce sperm, but with some hormone replacement therapy and things like that, they might be able. It's very iffy, but they may be able to get some sperm to be generated, so they may be able to pass, have children of their own. All right, let's go on to the next one. Okay, we're going to discuss a few more non-disjunction events. This is the last slide. So we've got poly X or triplo X. So this is our individuals that are XXX. So normally females have 2 X chromosomes, and these individuals, they have more. So there's not an extremely distinctive phenotype, except that they're usually taller and thinner than your typical normal female. Some may have delayed motor and language development. Some may have menstrual difficulties, but may menstruate regularly, but many are menstruate regularly and are fertile. So basically, these individuals again may have some difficulties, but they're more or less fairly typical of a female, except they're a little bit taller and thinner. Now, there are some females that have 4 copies of X chromosomes. These are very rare. They tend to, again, be very tall and thin. Now, at this point, they do tend to have some mental impairments. Okay, the last one I'm going to look at here is Jacob syndrome. These are XYY males, so they have an extra Y chromosome. Okay, these individuals, now they're actually they're usually taller. So, that's a little weird, but again, okay, so the androgens work strangely. So they might have early on, the androgens such as testosterone actually promote growth. So maybe perhaps that's what's happened here with these guys. They often suffer from persistent acne. They tend to have speech and reading abnormalities, but are otherwise normal. So about 1 in 100 males are affected. Now, there's typically no really major behavioral differences between XY and XXY, except there is a slight tendency to exhibit antisocial tendencies. So if you look, for example, in prisons, you'll find instead of 1 in 100 males, you might have a higher percentage of males in prison. So they do seem to exhibit some tendency toward these antisocial tendencies that would, behaviors, that would get them into jail. Okay, that's it for this section of the chapter. Thank you very much.